Hello, Bio 80. My name is Natalie,
I'm Anushka.
My name is Natalie Chaw.
I'm Nika.
And I'm Renee.
and our group's presentation is on botulism and Botox. For some context, botulism is a rare poisoning caused by toxins produced by the Clostridium botulinum bacteria. These toxins attack the body's nervous system, creating muscle weakness and paralysis in the body. In the worst case scenario, botulism can even cause death. Throughout this presentation, we will discuss both the dangers of the botulinum toxin as well as the modern day benefits of the toxin. Slide 2. So for a little background info, the botulinum disease was first recorded in 1735. Botulism actually gained its name from the Latin word for sausage. It acquired its name as in the late 18th century, several well-documented outbreaks of sausage poisoning prompted early botulinum toxin research. German medical officer Justinus Kerner published the first accurate and complete description of the symptoms of foodborne botulism by 1822. He did not succeed in identifying the toxin, which he called sausage poison. Kerner was also the first to envision using the toxin for therapeutic use after experimentation on animals. About 80 years after Kerner's work, the Clostridium botulinum bacteria was finally discovered by Emil Pierre van Ergenman, a distinguished professor of microbiology, around 1895. The discovery followed a botulism outbreak after a smoked ham dinner in Belgium. For a while, the toxin was only thought to contaminate meat or fish. However, in 1904, there was a botulism outbreak in Germany that occurred from canned white beans. In 1910, J. Lucius found that the canned white bean botulism was caused by a different strain of the toxin. In 1940, during World War II, Dr. Edward Shaw's finally isolated the crystalline form. Interestingly, military scientists experimented with the toxin in unsuccessful efforts to develop a weapon. Okay, slide 3. After the war, the military provided the toxin to interested scientists and researchers. By the early 1970s, scientist Alan Scott first injected the botulinum toxin to weaken the ocular muscles in monkeys in efforts to create crossed eyes. Interestingly, while testing the treatment on monkeys, Alan noted the skin between the monkeys' eyebrows had far reduced wrinkles. Then in 1977, with the FDA's approval, Alan Scott began cross-eyed treatments on humans, which was successful. Alan began working with ophthalmologist Jean Carruthers in 1982. As Jean began performing injections for eye spasms, eventually a patient approached her saying, "You didn't treat me here," and pointed between her brows. She continued to say that when you treat me here, I get this beautiful, untroubled expression. This encounter inspired Jean and her husband, Alistair Carruthers, a dermatologist, to pioneer the use of the botulinum toxin for cosmetic uses. And by 1991, they had 18 patients. Eventually, Alan, now considered the father of Botox, sold the rights to what later became known as Botox to Allergan, a pharmaceutical company, in 1991. Allergan developed Botox into a cosmetic treatment to reduce wrinkles and improve appearance. For a little FDA timeline, they approved the use of the toxin for therapeutic treatments in 1989, and in 2002, the FDA approved Botox for temporary aesthetic treatments for moderate to severe frown lines. Next. So Botox is now in 95 countries around the world. Over 100 million vials have been sold in the US since 2002. And there are over 500 peer-reviewed articles produced regarding Botox as a cosmetic. While Botox is mainly known for treating wrinkles, it can be used to treat a variety of other health conditions, including migraine, headaches, lazy eyes, twitchy eyes, urinary incontinence, acne, hair loss, and cervical dystonia, a painful involuntary neck spasm condition. Thank you.
Okay, so before we dive too deep into the underlying scientific basis, here's a general overview of the relationship between the bacteria we will be talking about today and its toxin. The specific bacteria at hand is the Clostridium botulinum, and this is the microbe that causes botulism. The connector between the Clostridium botulinum and botulism, believe it or not, is the same connector that makes the Clostridium botulinum effective in Botox's muscle paralysis and nerve blocking. This connector is the botulinum toxin, which is secreted by the Clostridium bacteria. You can go to the next slide. To begin, we can take a look at the structure of the Clostridium bacteria and what features it possesses that permits it to partake in the process of botulism or for Botox use as well. This bacteria is prokaryotic, hinting that it does not have a nucleus or a membrane to surround its organelles. Like unicellular prokaryotes, it evolves through binary fission. The process of binary fission is seen in the bottom image as one cell is splitting into two. This feature of the bacteria is beneficial because it means that it can reproduce extremely quickly. Another way in which the structure of the bacteria affects its viability is that it can go between a spore state or a vegetative state. You can go to the next slide. The spore state is better for survival in harsh conditions because it is dormant, yet the vegetative state proposes its uniqueness in that an aggregation of its kind is the only state to produce the neurotoxin of the botulinum toxin, which we will be talking about very shortly. To kill off this vegetative state, intense heats can be used for denaturation. You can go to the next slide. So now that we have taken a closer look at the Clostridium bacteria, let's dig deeper into the biological structure of the botulinum toxin and how the assembly of its DNA to its physique plays a role in its function. So when entering the body of any organism, the botulinum toxin must be physically fit to withstand the forces of the environment pushing it towards death. And one of these factors that would be problematic for the toxin survival is the gastrointestinal tract, which not only has a low pH, but also breaks down complex proteins into polypeptides or even further down into amino acids. In order to combat this negative situation that could hinder the viability of the toxin, it is immersed in protective proteins on the membrane. These protective proteins are part of a cloaking system that helps it withstand fatal conditions. And lastly, for this biological aspect, we can see that the toxin is made up of a heavy chain and a light chain. The heavy chain and light chain have distinctive rules that help them work together to enter the neuron. While the heavy chain has the translocation domain and the receptor binding domain, the light chain possesses the catalytic domain as shown in the diagram. So essentially, the light chain needs the heavy chain in order to bind to the neuron, and once it has bound, it is able to use its catalytic domain to its advantage. So now we can move on to the pathways, and you can go to the next slide in order to illustrate this. Again, while knowing the basic biology of both the Clostridium bacteria and the botulinum toxin is useful, it's important to glance at the pathway the toxin takes in order to cause paralysis, as seen in the use of Botox in modern day. To begin, the bacteria secretes the toxins in a double chain-like shape. This is the same set of chains that we talked about in the previous slide, and now it is being represented by the light blue and the dark blue blocks. Together, the heavy chain and the light chain are able to bind to the neuron, and they are able to enter through endocytosis. So, how does the contrast of a heavy and a light chain play a role in the effectiveness of the toxin? Well, the heavy chain is the alpha, and the light chain is able to follow it in. And once this first step of binding has been completed, the toxin makes its way into the neuron. From here, the light and the heavy chain go their separate ways, since the heavy chain has done its role in aiding the light chain into the cell. The light chain makes its way over to the snare proteins and splices them destructively using their function as proteases, which break down polypeptides. In simpler terms, the light chain, with the help of the heavy chain, has one, binded to the neuron, and two, entered via endocytosis. Moving through the cytosol of the neuron, the light chain reaches and splices the snare proteins, which are essential for neurotransmitter communications. An example of how the spliceness hinders communication is shown in the diagram. We see that acetylcholine is not released due to the cleaving of snare proteins, and acetylcholine is a neurotransmitter that regulates muscle contraction. This downfall of communication hinders the arousal of muscle cells, suggesting strict paralysis. All in all, the botulinum toxin enters the neuron and destroys the snare proteins, hindering muscle movements.
Okay, now we're going to move on to the ecological aspects of Clostridium botulinum. Next slide. So it is typically found in the natural environment in dust, soil, river water, or seawater, and it reproduces through its spores. And it mobilizes through utilizing surface waters in heavy rain or dust carried by wind. And the environment that it typically thrives in is one with low oxygen content, but it can also be found in toxin unaffected organisms. And this includes algae, plants, and invertebrates. And these organisms can actually serve as biotic reservoirs, which can turn to be toxic once decomposes. And while the bacteria itself is not harmful, it produces poisonous toxins when it is deprived of oxygen. This is why it's more harmful when it's contained. So some examples is inside human bodies, canned foods, closed bottles, and stagnant soil or mud. And this formation of toxin leads to an illness called botulism. Next slide. So there are ways to stop the production of toxins. So something that we should know is that the most favorable environment for the growth of the spores is typically 6.2 to 7.3, and the maximum pH for toxicity is 5.7. So knowing that the toxin is a protein, it can be denatured by heating. So by heating something that is decontaminated to a temperature that is equivalent to boiling, it will decontaminate and detoxify the affected product. Next slide. So in a situation where heat is not available, there are other ways that we can control the growth. And this includes drying the product, lowering the temperature, and increasing the acidity, and adding sugar or other chemicals. But some of these methods may not be as reliable as using heat, because it's really hard to kind of monitor the growth of the bacteria just by using these methods. But in humans, we can prevent toxin production in food, and that involves denaturing the toxin, and this is why we heat up our food before we eat. And if you look to the right, you can see a little picture about botulism safety in canned foods. So next time you look at canned products, make sure that it's not bulging, because if it is bulging, most likely that it is dangerous, and do not open it.
And now we're going to be talking about the Clostridium botulinum toxin in Botox. So it was first invented and used to treat muscle spasms, and in 1978, Alan B. Scott used the toxin to treat cross-eyed individuals and eye twitches, but instead it proved to improve wrinkles, and then it was first cosmetically used in 1990 to reduce facial wrinkles. When the botulinum toxin is injected into the skin, it blocks the nerve signals to the muscle where it was injected. So the muscle cannot contract if there is no signal. The toxin prevents the release of the neurotransmitter acetylcholine from the axon ending at the neurotransmitter junction, which causes flaccid paralysis. Each vial of Botox contains 100 units of the toxin. The 100 units are made up of Clostridium botulinum type A neurotoxin, 0.5 milligrams of albumin, and 0.9 milligrams of sodium chloride. Clostridium botulinum is a bacteria. So the bacteria in the 100 units, when it is injected, creates the toxin. The 100 units also contains 5 nanograms of Clostridium protein per vial. And 100 units is recommended because Clostridium botulinum type A neurotoxin is dangerous because it secretes poison. There are multiple dosages, like 200 units and up, but 100 units is recommended. Otherwise, it can be extremely harmful to the receiver because of the Clostridium botulinum type A neurotoxin. The dose of the toxin depends on the size of the muscle mass that the toxin is being injected to. So, a larger muscle mass equals a higher dosage, and vice versa, and higher dosages are more dangerous. Usually, when you get Botox, you need repeated injections for years. So, people who get higher dosages and frequent injections, that can lead to the creation of antibodies, meaning that Botox isn't working on them anymore. So, it is extremely recommended that you get low dose, infrequent injections in order to prevent botulism.
You can go to the next slide. Okay, to review, botulinum neurotoxins are a product of several species of bacteria belonging to the Clostridium genus. As a neurotoxin, they directly affect the body's nervous system, namely at its peripheral nerve terminals, inhibiting the release of acetylcholine. This neurotransmitter plays a vital role in the peripheral nervous system. Considering its range, for simplicity, nerve cells utilizing acetylcholine compromise what is called the cholinergic system. The first point of interest is in the autonomic nervous system, containing the sympathetic and parasympathetic subdivisions. Each one is responsible for visceral processes in the body, and each are a different side of the same coin. The sympathetic nervous system's role relates to the fight or flight state, while the parasympathetic nervous system's role relates to the rest and digest state. Together, they are responsible for either stimulating or inhibiting the same functions, such as heart rate, digestion, and respiration rate. Next slide. Acetylcholine also plays a big role at neuromuscular junctions in the somatic nervous system. This is where nerve endings innervate with skeletal muscle to send the signals necessary for muscles to contract. Simply put, acetylcholine is a major factor for enabling voluntary movement. Next slide. Botulinum neurotoxins essentially enter the neuron and paralyze it in a multi-step but rather complicated process, as described previously, ultimately preventing the release and use of acetylcholine. In doing this, it disrupts important functions like heart rate, respiration rate, and movement, leading to a rare but serious illness known as botulism. Now that we understand the underlying mechanisms, it can help us understand the effects of botulism as an illness. It begins as a weakness controlling the muscles in the eyes, face, mouth, and throat, marked by blurred vision, drooping eyelids, and trouble speaking. The weakness can spread to the neck, arms, torso, and legs, making movement difficult. But the real danger comes in how it affects respiration, making breathing difficult and requiring emergency care for breathing assistance. Botulism is rare, and only 5 to 10% of those inflicted find it fatal. But this mortality rate drastically increases if early diagnosis and immediate treatment are not given. The paralysis affecting skeletal muscles is reversible at a time, but can last anywhere from 2 weeks to 4 months. The paralysis affecting autonomic cholinergic nerve terminals can last up to 15 months. Next slide. There are 7 types of toxins, A through G, 4 of which cause botulism in humans, types A, B, E, and F. It comes in 3 common forms. Foodborne botulism is caused when the bacteria thrive and produce this toxin in a low oxygen environment, particularly in improperly sterilized canned meats and other low acid preserved foods. Botulism normally takes effect within 8 to 36 hours of eating contaminated food. The second form, wound botulism, is a result of the bacteria getting into a cut and creating an infection that produces a toxin. Those who inject heroin are at increased risk for this form. The third form is infant botulism. This occurs when babies between 2 to 8 months consume and grow the spores in their intestinal tract. They are most likely exposed to it through contaminated soil, but honey is suspected to be another source. Next slide. Thank you for listening, and here's our works cited.